Genetic mapping of genomes. Linkage analysis is the basis of genetic mapping. Recombination fraction is a measure of the distance between two loci. Two loci that show 1% recombination are defined as that they are being 1 centimorgan apart from each other. In other words, we can say that one map unit is equal to 1 centimorgan. Genetic mapping involves determining the location of genes on a chromosome relative to other genes using genetic crosses and pedigree analysis. As an example, genetic map of human genome has 24 different maps that include one for each of the autosome, genetic map of X chromosome, and genetic map of Y chromosome. Marker alleles in genetic crosses help to determine crossover rate between linked genes. This is a procedure that how genetic mapping of the genomes is performed. Individuals with different alleles at two or more loci are crossed and their offsprings examined. Most of the offsprings will have phenotype corresponding to linked genes. A few progeny will be recombinant. The frequency of the recombinant phenotype is calculated as a percentage of total offspring, giving the recombination frequency or the genetic distance. Experimental crosses are not done in case of humans. When we talk about the genetic mapping in humans, then experimental crosses are not done in humans. So genetic mapping relies on pedigree analysis and is limited by rarity of large multinational pedigrees showing segregation of defined linked traits. Usually, Lord score is method for used for statistical analysis of pedigree data in case of human genetic mapping. A Lord score usually compare the expected distribution of traits if they are linked and if they are not linked. Lord score is the log 10 of the ratios of two probabilities. Higher the Lord score, closer the two genes. The map distance for linked marker is computed from the recombination frequency. Mapped features that are not genes are called as DNA markers. A DNA marker must have at least two alleles to be considered as a useful genetic marker. Mostly three types of the genetic markers they are used in case of genetic mapping. They are RFLP, SSLP, and SNP. Genetic mapping of the genomes. To test linkage between the genes for two traits, certain types of the mating are examined, whether or not the pattern of combination of traits exhibited by the offsprings, that whether they follow the law of independent assortment. If not, then the gene pair for those traits must be linked. That is, they must be on the same chromosome pair. So in case of genetic mapping, whether we check that there is a linkage, mean the genes or the loci, they are linked or they are not linked. What are the recombination frequencies? Two genes that undergo independent assortment have 50% recombination frequency. It means that they are located on non-homologous chromosome. It means that they are unlinked to each other. While the genes with recombination frequency that is less than 50%, it means that they are on the same chromosome. In other words, we can say that they are linked with each other. Here is the example of the recombination frequency, that how recombination frequencies are calculated. Here there are two homologous chromosomes. There are 49 meiosis with no crossover. So it means 49 non-recombinants and all the 49 recombinants. But in addition, here we can see that there is one meiosis with a single crossover. It means this chromatid is non-recombinant, this is non-recombinant, and these two, they are recombinant. So they have exchanged their fragments, so they are recombinant. When we have to calculate the frequency of the recombination, then it means the recombinants, all the recombinants, 1 plus 1 over all the non-recombinants, that is 49 plus 49 plus 49 and plus 49, and all these recombinants and non-recombinants. So it will be 2 over 200, it means 1%. So 1% means that 1 map unit, in other words, it is 1 centimorgan. So the recombination frequency is 1 centimorgan. In other words, we can say that the allele or the gene A and B, they are apart from each other 1 centimorgan. So this is the method or the formula that how recombination frequencies are calculated. This is the formula for calculation of the recombinant frequency, number of the recombinant progeny over total number of the progeny multiplied by 100%. So in this way, we can calculate the recombinant frequency. Genetic mapping of the genomes is calculated by the recombination frequency that can be calculated in the form of Lord score. So here is the formula that how Lord score is calculated. Lord score greater than 3 is considered as evidence for the linkage, while on the other hand, Lord score less than -2 is considered that there is no linkage, in other words, to exclude the linkage. Genetic mapping of the genomes, linkage analysis is the basis of genetic mapping. Recombination frequency is a measure of the distance between two loci.